Still about evolution and descent with modification over a long period of time, but now we're going to start looking at pieces of evidence to support evolution and descent with modification. In Chapter 19, there are several main ideas of natural selection, so to recap them, please note that evolution is change in species over time, and it is a significant amount of time, not a year or 2 years. There is an overproduction of offspring, which leads to competition for resources, which is going to guide natural selection and survival of the fittest based on that competition. Heritable variations exist within a population, so when you have a group of the same species, there's going to be differences in their traits, and they vary. Those variations can result in differential reproductive success, which again is code for survival of the fittest. And over generations, this can result in changes in the genetic composition of the population. So if for some reason, again, maybe having blonde hair was a benefit or white fur is a benefit, over time, all that population is eventually, if white hair is best fit for the environment, over time that is going to be all the hair or the fur that we see in that population, and that would be a change in the genetic composition, excuse me. And then a final note, again, I said this, I think, on the last slide in 19.2, individuals do not evolve. Populations over an extremely long period of time evolve. There are 4 pieces of evidence for evolution that you will need to know and be able to provide examples for. One type of evidence for evolution is just direct observations. Another type is homology. Third is the fossil record. And finally, biogeography. And I'll get into these on the next few slides with what they are with examples. The first evidence for evolution is direct observations. So there's 3 examples that I'm going to briefly go into. The first is when you have an insect population become resistant to certain pesticides like DDT. So mosquitoes are resistant to DDT, but that wasn't the case when DDT was first invented. So initially, the DDT is sprayed in an area, mosquitoes die, but not all of them. And I don't know what gene it is that is responsible for certain mosquitoes staying alive, but there's a gene that the mosquito had when DDT was first sprayed. Those mosquitoes stayed alive. And because they're the only mosquitoes alive, they will then mate, lay their eggs, and then their offspring will have either that mutation or that gene. So as time goes on, natural selection has favored those mosquitoes that were resistant to the DDT, thus causing a change in that insect population. Another example is there are different kinds of pathogens which are disease-causing, either bacteria or viruses. There's certain types of bacteria that are antibiotic resistant, and one example is MRSA. So we have a strain of bacteria that lives all over us, on our skin, in our nasal cavity. It's a certain staph bacteria strain. Now, on its own, it's not harmful for us, but it has evolved in some cases into, which we probably have heard, a flesh-eating bacteria. That's what MRSA is. And the reason that MRSA is around is because that staph was originally treated, I think, with penicillin, then methicillin, and it built up resistance. And when it built up resistance, overall it became a lot harsher, a lot more dangerous of a strain of staph. And that, given its survival, again, natural selection favored for those bacteria that were resistant to survive and then pass on their genes to their offspring. A third example is the peppered moth. So two variations, and you can see these pictures in the bottom right. So the peppered moth living in the city versus the country. So the original version of the peppered moth is that white speckly version, as the name suggests. And when that peppered moth is in the countryside or in a forest, when it's on tree bark, you can't see it. So it has really good camouflage. Any of the peppered moths that had a mutation for being black or a gene for having black body with the black wings, it would get targeted or eaten by different birds or different animals. So over time in the countryside, the peppered moth that had genes for being speckled would survive. So in the country, natural selection is favoring the original tone of the peppered moth. But it was observed that in the city itself, when peppered moths were, whether it's up against a house or maybe even a car, just within the town itself, the speckled peppered moths were getting targeted and eaten by whatever, again, was hunting it, essentially. And the black peppered moths, were surviving. So it was easy to observe that based on the environment, natural selection is favoring the genes for either the peppered version in the country or the black version within the city. And those, again, are all examples of direct observations for evolution by the way of natural selection. The second piece of evidence for evolution is homology. And homology is the study of the characteristics in related species that have underlying similarities, even though their outside physical function may differ. So there's going to be several examples of homology. So one of them is called homologous structures, and these have similar anatomy from a common ancestor. So I'm just going to be very brief because I have another slide showing you this. The examples would be the forelimbs of humans versus cats versus whales or a dolphin and a bat. Embryonic homologies, this is going to be similar structures in early development. And examples would be vertebrate embryos with a tail in pharyngeal pouches. Another type is vestigial organs, and these are going to be structures that have little or no use at all to the organism, such as a flightless bird having wings. And molecular homology, this is similar DNA and amino acid sequences. All of these 4 examples I will go over on individual slides. Homologous structures are an example of homology. So comparing 4 different organisms, their forearm, if you will, at least in the terms of the human, on the external, physically, you would think that because these serve different purposes, they would be very anatomically different. But they are, in fact, very similar. So if we look at the bone structure inside, in the upper arm, you have the humerus, and everything's color-coded here. Then you have the radius and ulna together, the carpals, metacarpals into 4 or 5 digits, depending on the organism, being the phalanges. So these structures, again, the idea of this chapter is descent with modification, that there's a remodeling through time. So these structures make little sense on their own being so similar unless there was some sort of common ancestry. And please note that common ancestry does not mean that in the past there was a human cat or like a human whale swimming around or a human bat flying around. I suppose unless it was like Batman, but not realistically. One of my favorite Another example of hominology is embryonic development. So when we look at different organisms in different parts of their life, and in this case, during early development before either it hasn't been hatched or born, this would again be an indicator of some sort of ancestry. There's features here that are not visible in the adult, but visible in that embryo stage. So one example of that would be pharyngeal pouches. So in a chicken versus a human, these develop into, for at least humans, the ear and the throat area. If we were looking at an embryo of a fish egg, these pharyngeal pouches would turn into gills. In the chicken itself, it turns into some sort of a gland. And again, we can make these observations. And this is also important to note that how are those cells differentiated? Like, why do those cells turn into a gill in a fish and then turn into, the ear throat area for us? So these embryos are also not just the adult forms are studied, but also every stage of development. One of my favorite examples of homology is vestigial structures, because it really gives you an idea to kind of start thinking about evolution and how there's these structures that don't have any real use to the organism. So some of the examples would be, one, a flightless bird. Why does it have wings unless it's for flight? So that's one example. In snakes and in whales, there are bones in the lower region that are part of a pelvis or leg bones. And a whale, a snake, you don't need a pelvis unless you have legs, basically, which neither a whale nor a snake has. Other examples of vestigial structures, again, having no real use to the organism, would be, at least in us, an appendix. So this is a feature, part of our gastrointestinal tract, or like part of our lower digestive system, and it doesn't serve a purpose. Another example would be any sort of an organism that lives underground. So moles, some of these animals have like remnants of like eye sockets. Again, why would you need eye sockets unless you had eyes? And these organisms that live underground don't have the need for any sort of vision. The final example of homology, again, homology being characteristics in species that have differences, would be molecular homology in comparing the DNA and amino acid sequences. So when we look at the table, what this table represents is the number of amino acids that differ from a human hemoglobin polypeptide. So it's 146 amino acids. Again, hopefully we know amino acid, amino acid, amino acid, amino acid put together is a polypeptide chain. And when you get a lot of that together, that would be, at least in this case, what hemoglobin is, a type of protein. So when we look at humans in the 146 chain of amino acids, there isn't. It would be a difference of 0. But when we compare our number versus the rhesus monkey, who we are totally different than a monkey, there's only 8 amino acids, that's 8 molecules that are different in that hemoglobin. Comparing to a mouse, there's 27, a chicken, 45 different molecules or amino acids. A frog, it's 67, and in a lamprey, again, totally different, 125 of that 146. So, the entire 4 slides that I've been going over, I mentioned common ancestry. These are just examples of ways that when we look at all things that are alive, making like a compare and contrast list. Why are these things similar? Could they be related? Why are these things so different? Are they definitely not related on any level? So again, trying to crack the code of, what is life? Where did everything come from, and so on. These are just 4 different ways that scientists use with homology in comparing it to descent, or proving descent with modification.